Hello, everyone. Good morning, good afternoon, or good evening, depending where you are connected from. In this webinar, we will discuss about the opportunities for innovative radiopharmaceutical diagnostics and therapeutics discovery for cholangiocarcinoma with Allison Cohen. She's from the MD Anderson University of Texas. I will moderate this session. I'm Marco Rengo. I'm a radiologist, and I come from the University of Rome, Sapienza. So, Alison, I think we can start with your presentation.
Sure. Can everyone hear me? Yes, that's a yes. All right.
I can hear you.
Okay, good. Yeah, I guess they can't talk. So thank you for introducing. And as he said, I'll be talking about some opportunities for innovative radiopharmaceutical diagnostics and therapeutics discovery in cholangiocarcinoma. So just a brief outline of the talk. I'll start with a brief background and some rationale for positron emission tomography imaging, just for people who aren't imagers. This way, everyone's kind of on the same background level. And then in part one of the talk, I'll discuss a novel PET tracer that our group's been using for imaging of liver lesions. And then in part two, I'll discuss some future opportunities, including Theranostics and some new targets for imaging and therapy. Precision medicine is tailoring medical treatment to the individual characteristics of each patient. Individual tumors have unique cellular, molecular, and genetic characteristics, and we need to understand these characteristics in order to match patients with cancer to ideal therapies. Despite advances in techniques such as genomics, proteomics, and liquid biopsy, there remains room for improvement, and the role of molecular imaging in precision medicine is increasing. However, there are still critical gaps to consistently match patients with cancer and ideal therapies. And in our group, we hypothesize that PET imaging can help guide the path to precision cancer medicine. So there are a number of different advantages for PET imaging. The sensitive and quantitative nature of PET, coupled with the ability to produce targeted PET tracers, renders PET uniquely capable of detecting tumors and profiling their specific features. PET imaging provides a quantitative functional measure of tumor phenotype, and when it's coupled with tissue-based or liquid biopsy approaches, it can provide a greater breadth of biological characterization. Critically, the functional nature of PET allows a quantitative assay of biological processes non-invasively in vivo, such as the activity of transporters and enzymes, which are not directly measurable by other means, and this makes it complementary to approaches such as genomics. In addition, PET imaging provides these measures on a lesion-by-lesion basis, eliminating the ambiguity of inter- and intratumoral heterogeneity. So there are a number of different tracers that have been developed for cancer imaging, and on this slide, I just show a couple of different examples. In the top left, we have carbon-11 acetate, and this can image fatty acid uptake into tumors. On the top right, we have 18F4-fluoroglutamine, and this can image glutamine uptake into tumors. On the bottom left is fluoroestradiol, and this can image estrogen dependence. And on the bottom right is 18F-fluorothymidine, which can image proliferation. So at MD Anderson, our goal is to create molecules that allow for a number of different things. So we look to diagnose cancer, for staging and treatment planning, as well as for therapy. So on the next few slides, I'll just give a couple of examples from our work on each of these areas. So as I mentioned, PET can be used to improve diagnosis, and shown on this slide are representative images from 3 patients that have lung lesions that were determined by standard of care. But as you can see from these images, it's hard to determine whether these lesions are benign lesions or represent cancer. So using PET imaging with a novel PET tracer, our group determined that the patients on the left had benign lesions, whereas the patient on the right had cancer, as shown by the increased uptake of the PET tracer in this lesion. PET can also be used to complement drug development. So shown on this slide are representative images from a patient that was imaged pre and post-treatment with an estrogen receptor downregulator. And the post-treatment image was acquired at cycle 2, day 1, 4 hours after this patient took the drug. And we can see that there are multiple lesions in this pre-treatment image. And on the post-treatment image, we see that the drug is on target. So we've blocked the uptake of the tracer. And PET imaging can be used to visualize target engagement, as well as to help determine the optimal dosing for therapeutic regimens. So another example where we're using PET imaging in treatment response. So in this case, we're using PET to predict response to complex regimens of drugs earlier than the standard of care imaging. So our group is conducting a trial that's continuing at Vanderbilt, where we're imaging patients that are undergoing treatment with combined EGFR antibody inhibitor therapy, as well as a glutaminase inhibitor, CBA39. And we're performing glutamine PET imaging at baseline and after cycle 1 of therapy. And shown here are just 2 representative examples from our patients. On the top, we have a patient that did not respond to therapy. And as you can see on the post-treatment images, we see the presence of additional lesions following treatment. On the bottom, we have a patient who had a partial response. You can see a liver lesion in the pre-treatment image, and the uptake of the tracer goes to background levels, indicating that the treatment is working in this patient. So PET can also be used to match patients with therapy. So our group reported preclinical PET imaging using carbon-11 acetate, in this case in genetically engineered mouse models of hepatocellular carcinoma. And we're using carbon-11 acetate to reflect the activity of the acetyl-CoA synthetase enzyme ACSS2, which is the enzyme responsible for sequestering acetate to synthesize acetyl coenzyme A. So we tested carbon-11 acetate uptake and correlated it with ACSS2 expression in 2 different mouse models of liver cancer, the TAG and the c-Myc delta P10. And we observed in the wild-type mice, we see the expected biodistribution with modest uptake in the normal liver. We see significantly higher uptake in the livers that have the tumors. And we also, shown here, we have the time activity curves for the carbon-11 acetate uptake. And we can see in the later time points, we see that the c-Myc delta P10 mice have approximately 3 times higher uptake in the tumors compared to the normal liver. And if we look at the histology here, we see that this tumor shows high staining for ACSS2. Looking at the TAG mice, we see that these tumors have modestly higher uptake of carbon-11 acetate on the PET, and this corresponds to the lower ACSS2 levels seen in the histology. And these observations demonstrate a strong correlation between carbon-11 acetate uptake and ACSS2 expression, and thus carbon-11 acetate PET could reflect sensitivity to ACSS2 inhibitors. In addition, we've used carbon-11 acetate in patients with hepatocellular carcinoma, so translation of this to the clinic would be relatively simple. So where do we see PET going in the future? So PET imaging can be complementary to liquid biopsy approaches, as I mentioned earlier. And liquid biopsy has become increasingly popular in recent years, and it's used to detect tumor-derived molecular specimens, including circulating tumor DNA. Unlike imaging, liquid biopsy can be used for screening and surveillance, but it fails to provide precise disease localization and monitor the extent of disease or staging. So our idea is that we can use circulating tumor DNA to detect the presence of cancer in a screening setting, and then use this to trigger PET imaging with a number of different PET tracers that can then be used to localize the disease as well as determine staging for the patients. So in terms of PET tracers, our group likes to compare it to the number of stars in the universe. So there are a lot of stars in the universe, but the number of small molecules that are possible is decades magnitude greater. And if we look at the human genome, there are 2 × 10⁴ genes. So in theory, we can develop molecules that can target any protein of interest. So moving on to part 1 of my talk, I'll talk about a novel PET tracer that our group is using. So for several years now, our group has been interested in this PET tracer S4318F fluoropropyl L-glutamic acid, which we shorten as FSPG. and it's a glutamic acid derivative that's specifically taken up via the XCT transporter. So shown on this slide is just a schematic of the pathways that are involved in the tracer uptake. So in cancer, glutamine and glutamate are often major metabolic and anabolic substrates of tumor cells in parallel or even in lieu of glucose-derived metabolites. So glutamine is taken up via the ASCT2 transporter, and this can be metabolized to glutamate via the enzyme glutaminase. Glutamate can then be used to fuel the TCA cycle, or it can be exported in exchange for cysteine via the XCT transporter. And this provides precursors for glutathione biosynthesis and also regulation of redox potential. A constant supply of glutathione is critical for tumor cells to protect against the toxic accumulation of reactive oxygen species, and this is particularly important during therapeutic interventions. Thus, FSPG can be used as a biomarker to read out tumor redox status. So FSPG has been studied in a number of different clinical trials by our group and others. Shown here are representative images from our patients. So we've performed imaging in colorectal cancer, hepatocellular carcinoma, and lung cancer as shown here. So our group recently opened a clinical trial at MD Anderson where we're using FSPG to evaluate hepatocellular carcinoma as well as other liver lesions. And this is a collaborative project with a number of different investigators. So Dr. Flint is our nuclear medicine physician and she's PI of the trial. My PI is Dr. Charles Manning. I work in his group. We also work with Dr. Ahmed Kaseb, who's a GI medical oncologist, and Dr. Asif Rashid is our pathologist for this study. The study consists of 4 different patient cohorts. So cohorts A and B are hepatocellular carcinoma patients, and these patients will either undergo liver resection or orthotopic liver transplant. Cohort C is a benign liver lesion cohort, and cohort D includes malignant non-HCC liver lesions. And in all 4 cohorts, we'll be imaging with FSPG, and there's a number of different correlative biology measures that we're also looking at in this trial. So our group has published a pilot study of 11 patients with hepatocellular carcinoma using FSPG. And this table shows the demographics of the patients. And I won't go into full details due to time, but I want to point out that the FSPG tracer was able to detect 12 of the 16 HCC liver lesions that were identified by standard of care imaging. And FSPG showed significant accumulation in the tumors compared to background liver. So the SUV values, which is a standardized uptake value for the background liver, was significantly lower than the uptake in tumors. And the low background is a major contributor to the promising tumor to liver background ratio, and this will be important for cholangiocarcinoma as well. So shown here is just a representative image from one of the patients on this trial. This patient has the typical biodistribution pattern for FSPG with activity in the oral cavity, the pancreas, and clearance through the kidneys with urinary excretion. So shown here is the maximum intensity projection image from the skull to the mid-thigh of this patient. And we see 2 areas of uptake of FSPG in the liver and modest background accumulation. Shown in panel B are the axial PET-CT fusion images that correspond to these 2 lesions. And we can see this lesion is a large liver lesion as identified by the standard of care MRI, and it's easily detectable on the FSPG. But importantly, we also detect a smaller lesion that's easily visible on the FSPG PET, but it's less obvious on the standard of care MRI. And liver explant analysis following resection confirmed that both of these lesions were HCC. And the detection of the smaller lesion, which wasn't as easily detectable on the MRI, indicates that FSPG PET may be useful for detection of small tumors that aren't easily seen by standard of care imaging techniques. So as part of the trial, I mentioned we have a cohort that is looking at malignant non-HCC liver lesions. And we recently got pilot funding to perform a small study in 8 patients with non-resectable intrahepatic cholangiocarcinoma. And this is done in collaboration with Dr. Cohen, who's a radiation oncologist at MD Anderson. And the aims of this study are to correlate baseline accumulation of FSPG in intrahepatic cholangiocarcinoma patients with clinical response to radiation treatment, as well as to compare the baseline accumulation in patients that express wild type and mutant BAP1. And since it's a pilot study, the goal is just to determine the feasibility of PET imaging to visualize these tumors and to predict their response to radiation treatment. So going back to our pathway, this one's a slightly modified version, but we still have FSPG is transported via the XCT transporter into the cells. And this XCT transporter exchanges glutamate and cystine. Intracellularly, cystine is reduced to cysteine. And again, this is the rate-limiting precursor for glutathione biosynthesis. And reduced glutathione plays an important role in redox maintenance for the cell and also helps protect against ferroptosis, which is a non-apoptotic form of cell death. So glutathione peroxidase 4, GPX4, uses glutathione to convert lipid hydroperoxides to lipid alcohols, and this suppresses ferroptosis. So as I mentioned previously, FSPG can be used as a non-invasive marker of the tumor metabolism and also the redox potential of the cells. So just to go into some of the background for the pilot clinical study, chromosomal aberrations and mutations in tumor suppressor and other genes play a role in cholangiocarcinoma, and one of the genes that is often deleted or mutated, particularly in intrahepatic cholangiocarcinoma, is BAP1. BAP1 is a deubiquitinating enzyme that acts as a tumor suppressor, regulating many cancer-associated pathways, such as cell proliferation, cellular differentiation, and DNA repair. And BAP1 also has a role in multiple metabolic pathways and in metabolic stress management. At MD Anderson, it's been shown that a BAP1 target gene is SLC7A11, which is the gene that encodes the XCT transporter, which is responsible for the FSPG uptake. So in normal healthy tissue, BAP1 negatively regulates SLC7A11 expression, and this controls glutamate and cystine levels and maintains cellular function. And since cystine is the precursor of glutathione biosynthesis, BAP1 also indirectly regulates the redox potential of the cell. Upon BAP1 inactivation through mutations, SLC7A11 expression is upregulated, resulting in a constant supply of glutathione and regulation of redox potential. And again, the redox maintenance is critical for tumor cells and provides protection against toxic accumulation of reactive oxygen species. So a constant supply of glutathione can enhance tumor survival, particularly during therapeutic interventions, suggesting a role for SLC7A11 transporter activity, as well as BAP1 mutational status in glutathione-mediated resistance to therapy. So given the relationship between BAP1 and SLC7A11, we hypothesized that intrahepatic cholangiocarcinoma tumors bearing BAP1 mutations will exhibit elevated expression of the XCT transporter and thus increased baseline FSPG accumulation, and that this will be predictive of tumor aggressiveness and radiation resistance. So just shown here is our study timelines and procedures. So all the patients on this trial will undergo standard of care, either CT or MR, within 4 weeks of the investigational PET imaging. The PET imaging is performed at baseline prior to radiation treatment. And we acquire a whole body FSPG PET CT scan. And this is 60 minutes after intravenous injection of the tracer. We also acquire a whole body transmission CT scan for anatomic localization and attenuation correction. The patients are treated with radiation as part of their standard of care, and we monitor treatment response using the follow-up standard of care CTs or MRIs. We are measuring a couple of different outcome measures as part of this trial. So we look at the standardized uptake values for FSPG in both the tumor lesions as well as the background liver, the number of lesions, as well as the change in tumor size, which is measured at pretreatment and after radiation therapy using the standard of care images. We'll look at the BAP1 mutation status, and also if biopsy tissue is available, we'll look at immunohistochemistry for a number of markers, including the XCT transporter as well as CD44. So far, we've imaged 3 patients on this trial, and we're still actively recruiting additional patients. So just as a little teaser, because we don't have full data analysis yet, but this is our first patient that we imaged with FSPG, and we believe it's the first patient with cholangiocarcinoma that's been imaged with FSPG. This patient had a poorly differentiated cholangiocarcinoma, and the patient was scheduled to receive radiation to the primary tumor. We see normal physiological radiotracer uptake. You can see the kidneys and urinary excretion of the tracer, as well as the pancreas in this patient. The background liver has a maximum standardized uptake value of 3, And as you can see, we see a large FSPG avid lesion that's in the liver. And this lesion had an SUV of 6, making the tumor to background ratio approximately 2 for this patient. So moving on to part 2, unless there are any questions on part 1.
There are no questions for the moment from the audience. I have just one question for you, just one quick question. Did you investigate the difference in accuracy between the FSPG and the FDG?
So we don't.
For the detection.
Yeah, so for cholangiocarcinoma, we're not acquiring FDGs. If they're available as part of the standard of care, we could look at that. For the hepatocellular carcinoma patients, we do have a cohort that will be getting the FDG as comparison. But since that cholangiocarcinoma is just a pilot study, we're not acquiring the FDG as part of the trial.
Okay. And in general, for the other cancers?
So hepatocellular carcinoma, we are acquiring FDG in a cohort of the patients, and we'll compare. So for the hepatocellular carcinoma, we're comparing FSPG to FDG and acetate as well. And we're getting pathology, and it's a larger study. That one's funded by an R01, so we can play a lot more with the tracers. The cholangiocarcinoma is just a pilot study right now, so the goal is to eventually expand. If the results look promising, we'll expand to a larger trial, and we can look at more things like ctDNA, FDG, other correlates to the PET tracer.
Okay. I think that there is a question. Just 1 minute. I check the for the enrolled patient in the CCA arm. Do you allow patient who previously received chemotherapy or only the no patient? So are patients treated with chemotherapy allowed in your CCA trial?
So these patients will be getting radiation treatment. They can be pre-treated before they enroll on the study. But there's usually a period in between when they receive their last treatment before they start the radiation.
Okay, the standard is just radiation, is not chemotherapy.
Yeah.
Okay, perfect.
Okay. So moving on to part 2.
I think you,
Is there another question or?
No. Sorry.
No, you're fine.
You can go on.
Thank you. So moving on to part 2, I'll talk about some future opportunities, including the use of Theranostics and some new targets that are relevant in cholangiocarcinoma. So just brief introduction. So what are Theranostics? Theranostics are radiopharmaceuticals that combine diagnosis and therapy. And this isn't the new concept. It's been around since the 1950s. But in recent years, Theranostics have become increasingly popular. And this includes the use of targeting peptides as well as antibodies in a number of different cancer types. So just to kind of walk you through the pathway for how we develop a radiopharmaceutical for Theranostics. First, we identify a target that we would like to develop a molecule for, and we perform chemistry to synthesize molecules that we think can target based on the computational modeling shown here. We then perform biology to select leads, so we can do ligand receptor binding assays, live cell imaging, and mass spectrometry proteomics. Once we have leads, we can move on to radiopharmaceutical development. So develop the chemistry to label the molecules with an isotope. And in this case, we're showing fluorine-18. And then move on to preclinical studies in mice, as well as ultimately, hopefully, clinical translation. So as I mentioned, there are a lot of different studies that are involved, including chemistry, structural biology, preclinical, clinical studies. So this requires integration of a large number of programs and a multidisciplinary team. So at MD Anderson, we're developing a Theranostics program that will have all of these different resources and the benefit of being at a large institution that has experts in all these different areas. So we have cancer biology and targets, chemistry, preclinical evaluation, radiochemistry, as well as clinical trials, and eventually, hopefully, standard of care clinical use. So shown here is just our research pipeline. And as you can see, we have, as I mentioned, we have chemistry, targets, translation, radiochemistry. But within each of these, we also have different sub areas of expertise shown by the smaller circles. So this requires quite a large group of people, a lot of different experts in a lot of different areas. But the ultimate goal is to be able to deliver the next generation of Theranostics to the clinic. So opportunities for Theranostics, there are a lot of different targets. So shown here are just some common targets in cancer. And some of them have tracers already developed for them. Others are more novel. And I'll discuss a couple of examples in the next few slides that have been used in cholangiocarcinoma. So starting with prostate-specific membrane antigen. So gallium-68 labeled prostate-specific membrane antigen, or PSMA, is a radiopharmaceutical tracer that has become increasingly promising in the staging and detection of recurrence in prostate cancer. But PSMA is also expressed in the endothelium of neovasculature for various other solid malignancies, including breast, renal cell carcinoma, and glioblastoma, colorectal cancer, non-small cell lung cancer, and hepatocellular carcinoma. And shown on this slide are representative images from a patient with prostate cancer. And these are gallium-68 PSMA PET images that quantify the disease burden pre and post-treatment in these patients. And as you can see from the title of the slide, there's also a therapeutic agent, which is labeled with lutetium-177 for therapy of prostate cancer. And the survival curves, you can see there's increased survival following treatment with this agent. So in terms of cholangiocarcinoma, a pilot case study was published in which they looked at PSMA PET in a patient that had metastatic cholangiocarcinoma. This patient had already exhausted all approved conventional therapeutic options, so they decided to image the patient with gallium-68 PSMA to assess PSMA expression and to potentially guide the radionuclide therapy, as I showed on the previous slide, so if these lesions show PSMA expression. In panel A is the maximum intensity projection image using the gallium-68 PSMA PET, and in panels B and C, we see the transaxial view as well as the corresponding non-contrast enhanced CT and the overlay of the PET CT. We can see in panels B and C that the gallium-68 PSMA identified liver metastases in these images. So the arrows point to the liver lesions. In panel D, we see the PET CT that shows nodal lesions in this patient, and panels E and F show bone lesions in this patient. So this patient has metastatic hepatic, nodal, and bone lesions that all demonstrate PSMA expression. And this indicates that this patient may potentially be eligible for radionuclide therapy using a PSMA-targeted radiopharmaceutical. So the conclusion was that PSMA-targeted radioligand therapies could be used in other cancers in addition to prostate cancer. So moving on to another target. So as all of you, I'm sure, know, cancer tumors consist of more than just the cancer cells. And there's a large variety of different cell types in the tumor, including immune cells. And for the radiopharmaceutical I'll be discussing next, it's specifically targeting cancer-associated fibroblasts. So cancer-associated fibroblasts are essential for tumor cell proliferation and metastasis. And a protein called fibroblast activation protein, or FAP, is overexpressed by these cancer-associated fibroblasts in several different tumors. And shown on the right are different FAP inhibitors that have been developed for studying fibroblast activation in oncology. And these inhibitors have been labeled with gallium-68 for PET imaging and have shown promising preclinical and clinical results. So a recent study quantified the tumor uptake of gallium-68 labeled fappy PET in various different primary and metastatic tumors to identify the most promising indications for future applications. And 80 patients with 28 different tumor types were evaluated. So as you can see, the representative images show a large number of cancer types, and they also quantify the standardized uptake value for each of these tumors. And cholangiocarcinoma is one of the tumors that showed the highest uptake of this tracer. And you can see the visualization here. Cholangiocarcinoma is a particularly desmoplastic stroma-rich tumor, and it's been shown that cancer-associated fibroblasts actually often outnumber the cholangiocarcinoma cells in these tumors, demonstrating the potential of the fappy tracer since it targets the cancer-associated fibroblasts. So shown here is a representative image of a cholangiocarcinoma patient that has elevated uptake of the fappy PET tracer. And we see pathology shows that this patient has poorly differentiated intrahepatic cholangiocarcinoma. And this group also performed immunohistochemistry and showed high FAP expression in the stromal cells, indicating the correlation between the fappy PET and the elevated FAP expression in the tumors. Several studies have also compared fappy PET to FDG PET. So shown here is a patient with primary intrahepatic cholangiocarcinoma, and we can see that the fappy PET has significantly higher activity as well as the higher tumor to background ratio compared with the FDG in this patient. We see two strong areas of uptake on the view here, and this corresponded to the two lesions seen on the CT. And the biopsy demonstrates this is a primary intrahepatic cholangiocarcinoma. You can see on the FDG, you can barely make out the tumors on these images.
Yeah, Allison, sorry. Yeah, the problem with the cholangiocarcinoma is that FDG sometimes is not diagnostic because the uptake is not always high. Yeah. So that's very interesting. Yeah, just a quick comment. You can
no problem. Yeah, no, this tracer actually, I was going to say, I'll show some more examples. It compares really well with, I was going to say, in terms of higher uptake than FDG.
Yeah, I mean, this can also increase the accuracy for the detection of smaller lesions, because with PET, we have some sort of dimensional cutoff, but it depends mostly from the uptake, because sometimes small lesion with the uptakes are not. So I think that this level of signal is also helpful to overcome the limit of small lesions.
Yeah.
It's very nice.
So shown here is just another example where we have fappy PET CT showing higher activity and higher target to background ratio than the FDG. So this patient was a treatment naive patient, and the PET was performed for initial staging. So in panel A, we have the maximum intensity projection image. Panel C is the fused PET CT image. E just shows the attenuation correction PET image. And panel B shows the FDG PET, the corresponding PET CT for the FDG, as well as the attenuation correction image for the FDG. And you can see again, the fappy PET shows higher uptake than the FDG. And the lesions were again confirmed using standard of care imaging, so MR images in this case. So this tracer has also been evaluated in comparison to FDG for local recurrence and detection of residual tumor lesions post-treatment. So fappy PET was shown to detect a larger number of lesions than the FDG PET. So shown here, we have a patient who underwent hepatectomy and chemotherapy. And the patient underwent restaging scans. So the fappy PET is shown in panel B. This is the whole body image. And we see the MRI that corresponds to this, as well as the PET MRI overlay here. And we see two lesions of uptake that likely correspond to liver metastases in this patient. The FDG initially does not show any uptake, as you can see in panel A, This patient then underwent reevaluation of disease 3 months later. And we see in panel D, the fappy PET, again, the gadolinium enhanced MR and the PET MR overlay. And in this case, we can see that the fappy avid nodules that were seen initially have increased in size and intensity on the follow-up scan. And the FDG that corresponds to this image is shown here. And now we see that there are 2 lesions here on the FDG image that confirm the diagnosis. And it was seen obviously 3 months earlier with the fappy PET. So this indicates the potential of this tracer to detect lesions earlier than the FDG. And again, to your point, smaller lesions. So fappy PET can also affect patient management. So in this case, they were using the fappy PET to detect residual tumor. So this patient underwent chemotherapy and then underwent PET MR for restaging. So the fappy PET is shown in panel A, and we have the PET CT overlay in C, as well as the corresponding gadolinium enhanced MR in E, and the fappy PET MR overlay shown in F. We see a liver mass with intense uptake that corresponds to the mass that's seen on the MRI. The FDG is shown in the whole body, is shown in panel B, as well as the PET CT overlay in D. And as you can see, the FDG PET did not visualize the tumor in this patient, and this could obviously affect patient management. So shown here is a patient where they were looking for intrahepatic metastases with fappy PET compared to FDG. And the fappy PET was found to have a better per lesion detection rate than the FDG. So this patient underwent postoperative PET CT for restaging. And the fappy PET shows numerous lesions, as you can see in panel A, as well as in the axial image in panel B. The CT scan also shows an ill-defined nodule in the liver. However, the FDG shows no abnormal uptake, as you can see in panels B and D. Pathology confirmed that this was actually a metastatic cholangiocarcinoma. So as I mentioned, this obviously affects patient management because underestimating the number of metastases can affect the treatment of these patients as well as staging. So finally, just an example of distant metastases. So fappy has also been compared to FDG in the case of distant metastatic lesions. And fappy was able to detect more lesions than the FDG. So this is an example where a patient has a metastatic brain lesion. So we have the fappy PET on the left, the FDG PET on the right. And you can see that the fappy PET detected a brain lesion in this patient that was ultimately confirmed by MR. However, this lesion is not detectable on the FDG PET. And again, this can affect the patient's management, their quality of life, and also their prognosis. So as I mentioned, fappy is used as a theranostic. So in addition to the gallium-68 label for fappy, it's also been labeled with lutetium for treatment. So shown here are representative images from a patient that had bone metastases. And we have fappy PET CT images in panel A, and also the whole body scintigraphy at multiple different time points post-injection of the lutetium labeled fappy. And these demonstrate uptake and retention in the metastases of this patient. In F, we see a post-treatment image taken 8 weeks after treatment, and this demonstrates a mixed response in this patient. And the patient ultimately had progressive disease, as you can see by new metastases in panel J. So one last example. So this is a preclinical study that used L1 cell adhesion molecule, L1-CAM, which has been known as a novel prognostic marker and therapeutic target for cholangiocarcinoma. And this study aimed to evaluate the feasibility of immunopet imaging and radioimmunotherapy using a radio-labeled anti-L1-CAM antibody, and they used cholangiocarcinoma xenograft models. So L1-CAM is expressed in cholangiocarcinoma, and it's also involved in tumor progression, migration, and resistance to apoptosis. And previous reports have shown that it could serve as a useful diagnostic marker with the potential to serve as a therapeutic target. There's also an antibody, a chimeric antibody, that targets L1-CAM with high affinity and selectively binds to L1-CAM positive cells. So in this paper, they conjugated this antibody to a chelator, NODA, and then labeled it with either copper-64 for PET imaging or lutetium-177 for SPECT or therapeutic. And this strategy could be used to stratify the patients based on the PET imaging and then treat the appropriate patients with the radioimmunotherapy. So shown here are representative PET images using the copper-64 labeled antibody. And we have various different xenograft models. And they were able to show that the PET imaging corresponds to the expression level that was found for the L1-CAM in these different xenograft models. So you can see low expression for the ACHN model with increasing expression as you go across. They also performed a blocking study. So they used cold antibody, pretreated with cold antibody, and you can see a decrease in the signal. And this was confirmed using digital whole body autoradiography. So these results demonstrate the potential of the copper-64 labeled antibody as an immunopet imaging agent that can be used for non-invasive and quantitative in vivo imaging of L1-CAM expression in the cholangiocarcinoma models. As I mentioned, this group also labeled the antibody with lutetium-177. So lutetium-177 also emits gamma and beta particles, which can be imaged using SPECT. So shown here are SPECT images taken at various time points post-injection of the 177 lutetium labeled radioimmunotherapy. And we can see that the tracer accumulates in the tumor and has relatively low uptake in the rest of the body. And this was retained up to 14 days. And you can also see, based on the size of so the dash shows where the tumor is, that this tumor increased in size based on the therapeutic effect of this antibody. And again, they confirmed the uptake using digital whole body autoradiography.
Allison, this is extremely interesting because in this way you can monitor in real time the effect of the therapy. Yeah. But it's not for all the radioactive molecule you're using, no? Because not all can be imaged.
So lutetium-177 is used, it's pretty commonly used for the radiotherapeutics. So it can be used to label, I was going to say it's been used to label, the PSMA was lutetium-177. This one's, I was going to say there are a number of different molecules that are labeled with lutetium-177, and you can do SPECT as well as the therapy with lutetium-177. Not all the isotopes can do that now.
That's very, very interesting. Yeah. Thanks.
So as I mentioned, it's also a therapeutic. So to evaluate the therapeutic effect, they use the lutetium labeled antibody in a xenograft model. So saline they used as a control. They also used just the antibody alone. And then they have an isotype antibody that was labeled with the lutetium-177 just to evaluate the effect of the isotope. So as you can see, the antibody alone didn't have any therapeutic effect. The isotype control, we do see a slight decrease due to nonspecific targeting. However, the tumors do still grow over the course of the experiment. However, if you look at the lutetium-177 labeled specific antibody, you can see that the tumors actually show a decrease in tumor volume across the course of the imaging. So there's a significant difference starting at day 10 from the vehicle treated group. But you do see eventually a 50% reduction from the initial tumor volume at day 27 here. In addition, they showed that this treatment was relatively safe, so there's no difference in the body weight of the mice over the course of the experiment. So conclusions. So PET imaging offers great potential to advance precision medicine. I hope I've shown you that PET can be used for diagnosis, staging, and evaluation of treatment response in various different cancers. The development of tracers targeting many proteins and pathways are possible. At MD Anderson, we've opened a pilot clinical trial that's being performed to evaluate the potential of non-invasive PET imaging with FSPG to visualize intrahepatic cholangiocarcinomas and predict their response to radiation treatment. And results of this trial could lead to improved clinical management of patients with intrahepatic cholangiocarcinoma. Theranostics offer the possibility of combining imaging and treatment, and there are several different theranostic agents that have been tested clinically, and many more are currently in development. So I'd like to acknowledge my group. My PI, I work for Dr. Charles Manning at MD Anderson. We have a large group of people here, as well as a number of different collaborators. Dr. Flint is our nuke med physician. Dr. Cohen is our radiation oncologist that we work with. Dr. Wei is our statistician. I'm part of the Department of Cancer Systems Imaging, which is part of the Division of Diagnostic Imaging. Obviously, we thank the patients and their families for participation on our trials. There are a number of different core facilities that we use to help with our trials for production of the radio tracer and imaging of the patients, our funding sources. And thank you for your attention. I'd be happy to take any other questions. And I also have my email there in case you want to contact me afterwards.
Thank you very much, Allison. Extremely interesting presentation. I learned something I didn't know. Since I'm a radiologist, I don't deal with the PET usually. I have some general question about this topic. So the first is, how long does it take for your research to be introduced in general in the clinical practice? Now you are studying, you have your research. How long does it take, more or less, for the use, the clinical use?
Right. So it depends on what you're labeling. So like if the antibodies, so like there are examples where the antibodies already used clinically. So like people have labeled EGFR antibodies with zirconium and other isotopes. If the antibody is already approved for use, then it's a lot easier to cross that barrier because you're just labeling it with an isotope. So you have, I mean, you still have to show safety, but since the antibody is already used clinically, it's a lot easier to do. If you're starting from scratch with a new ligand, it takes a lot longer. And I haven't been involved in the full process. The stuff that we have preclinically has not gotten to the clinic yet. But I would guess probably at least 10, 15 years if you're starting from the beginning.
Okay, that's what I was thinking.
I mean, you obviously have to, you develop the tracer, you have to validate it in vitro first, and then in preclinical studies before you can. Okay,
but starting from the already approved antibodies, it's easier. And for the tracer, is there a link?
I mean, if they, I was going to say, it can work for small molecules too. If there's a small molecule drug you can label, if it's already like a small molecule, like there are EGFR inhibitors that are small molecules, you can label those. And that's also if they're already approved. If the targeting moiety is already approved clinically, I think the path is shorter.
Okay, we have a question. Is there any information about fappy PET's rule in the early predicting tumor response to FGFR2 and IDH1 inhibitors in cholangiocarcinoma? So the rule of fappy PET in the early prediction of the response of cholangiocarcinoma expressing FGFR2 and IDH1.
So I don't know. I was going to say the couple of papers that I found are the ones that I presented. I don't know if anyone specifically looked at certain populations of cholangiocarcinoma. I think these were more open trials with patients that have a bunch of different types.
So we don't know yet.
Yes.
And another question I'm really interested in as a radiologist. Do you think there is any room for PET MRI in this? Do you think it will improve the accuracy or not?
So I think
because there are
PET MRI has a lot of potential for certain cancer types, and cholangiocarcinoma may be one of them. Yes.
Okay, because as a standard of care, MRI is for cholangiocarcinoma, it's you use it as reference standard for detection.
Yeah.
So I guess that it may, for liver, for example, it's better, should be better.
Yeah, I was gonna say
But
the PET MR, like the combined instruments are developing. I don't know that they've been fully optimized yet, but like you can do an MR and then you can do a PET and register those images, but they have instruments that can do both. And those I think are still kind of under development, but in terms of getting the best PET signal and MR signal.
Yeah, the optimization is difficult. Is extremely difficult.
But I think the potential for it is very good for cholangiocarcinoma.
Yeah, for sure. We have another question from the audience. Is fappy PET used to detect residual disease after surgery?
Yes.
The question, yes, is it used?
So in the clinical trial, it was evaluated for that. I don't remember what, I think it might have just gotten FDA approval, but I don't remember for what indications. But in the clinical trial, yes, they evaluated it for detection of residual tumor.
Okay, and then there is the second part of the question, very interesting. Does the time matter, since after surgery there is also a fibroblastic reaction that may cause false positive?
Yes, so one of the issues with fappy PET is that inflammation also could show up. So that's one of the reasons also that the PET MRI may be really useful, because the MRI could detect differences in terms of whether it's an inflammation related response. But fappy does have some potential for false positive results in inflammation.
So you have to wait sometimes after surgery to use the...
I don't know if the optimal time has been decided yet, but...
Okay, but it's not an immediate evaluation after surgery?
I don't think so, no.
Okay. And for the therapeutic effect, the safety profile we see in your slides in that population was good, but I mean the therapeutic effect is correlated to the, I think, with the accumulation of the tracer. Is it okay? So is the effect?
This one you mean?
Yeah, I mean, this is the you show that the safety profile is good. It was on mice.
Yeah, it was on mice.
Okay, it was mice, and but the labeling antibodies are not 100% specific for the tumor. So I guess that some molecule will go also in healthy tissues. Is it correct?
So you see, I mean, on the initial image, you see there's some, but over the course of time, like it clears out. You don't really see too much accumulation in the normal tissues in this, at least in this example.
Okay, so the effect depends from the concentration and time. Is it correct? So that's why it is relatively safe.
Yes.
Okay. And you show us in one of the previous slides that there is also a normal accumulation of the tracer in the oral mucosa and in the pancreas?
For FSPG.
Yes. For the FSPG, yes. And,
that one's not therapeutic though, so that one's just for imaging.
Okay. Yeah, okay, perfect.
So it can't be used for pancreatic primary pancreatic cancers. It can't be used for, but it has been tested for metastatic pancreatic cancers. But yeah, it's also not a therapeutic. So for imaging, you can get away with some normal accumulation. That's one of the
Okay.
I guess, challenges of developing a theranostic agent is you also have to consider the normal accumulation, because if you're conjugating it to a therapeutic molecule, you obviously don't want too much damage to normal tissue.
Yeah, correct. And for the diagnostic aspect, do you think that those kind of labeling antibodies or target molecules with contrast media developed for MRI may have a rule? As a radiologist, I'm used to change the contrast media, for example, hepatospecific contrast media, for detecting diseases, especially for liver disease. And in the past, I used also the ferrocarbotran, which is the micro iron molecule. We were using for liver imaging. So do you think that maybe room for labeling iron small particles with antibodies and use MRI instead of PET? Something like that?
You could, but the sensitivity of MRI is much lower. So the concentration of tracer that you would need to accumulate in the tumor. Huh?
Yeah, you mean as the signal to noise ratio.
So in order to get the signal on the MRI, you need more accumulation, like a higher concentration in the tumor with the MRI contrast agents.
Okay, so that's the limit.
Yes. But you could target MRI contrast agents with these antibodies too. It's just, I think you need, with the PET, you don't need as much of it to accumulate there. Does that make sense?
Yeah, absolutely.
I'm thinking of it like as in concentration. So like you have the tumor as, whatever 1 ml volume, you need to accumulate much more of it to get an MR signal than you would for PET.
Yeah, maybe with the iron and very high magnetic field scanners, 3, 4, 5, or 7 Tesla, maybe, yeah, you need less contrast because
so I mean,
if
you can do, I was gonna say, I guess I'm thinking here, our clinical scanners are usually 1.5 T, maybe 3 T, but we don't have like 7 T we have for mice, but we don't have a 7 T scanner for the patients.
Yeah, it will take a long time to do something like that. Because the advantage with MRI is that you are, it's radiation-free technique. So
yeah,
this field is better.
I was gonna say for PET, we're administering such small amounts. We always think, I mean, we test the safety, but we're administering such small amounts that we don't think of it as like we have for our radiation safety. We usually compare it to like if you do transatlantic flights, you're getting radiation from flying over too. So we usually compare it to like the number of transatlantic flights you take, and it's not usually too bad.
So radiation exposure is not an issue for the
PET imaging. I mean, if you're doing therapeutics, you're going to get a higher radiation dose.
Yeah, of course. Yeah, that's the goal.
Yes.
I think that we don't have any other questions. Okay, is there any other, so now you're, is there any other project you can give us some insight on these tracers?
So the only project our group has in cholangiocarcinoma is the one that I told you about. We have a lot of clinical trials looking at glutamine metabolism. So we have FSPG in a number of different trials. Hopefully there'll be some publications soon, but I was gonna say we're looking, I mean, I mentioned the hepatocellular carcinoma. We have a colorectal cancer trial. We have head and neck cancers. Yeah, I was gonna say we do a lot of PET imaging in our group. So the therapeutics are just coming up. So we're developing the program. But in terms of PET imaging, we do a lot of tracers and a lot of different cancer types. So anyone who wants to collaborate, the physicians can feel free to reach out. Yeah.
Okay, thank you very much. I think that we finish our time. So I hope everyone enjoyed the meeting, and hope to see you next meeting. Bye. Thanks very much, Allison.
Thank you.